Okay, welcome back, everyone. Hope everyone had a nice little break there. Up next on the program is just listed as Noxious Weed Update. Basically, when I put this program together, didn't know if we could actually advertise the name of the individual who would be giving this update. And so many of you remember Dr. Rod Lim when he retired, and I'll say, but the behind the scenes thing, when he retired, his position retired with him. So we actually created a new position that's noxious weed control in cropland and in non-cropland. And so as many of you know, something like Palmer Amaranth is on the noxious weed list in North Dakota now. So this individual will be able to evaluate some additional things, go above and beyond kind of what Rod looked at in non-cropland areas. So with that, it's my pleasure to introduce the person who accepted this position and will be starting later this spring, soon to be Dr. Quincy Law, who is finishing up his degree at Purdue University. So Quincy, if you're ready to go, we're going to turn it over to you.
Thanks, Joe. And hello, everybody. Had met a few of you when I first visited North Dakota. I'm currently still at Purdue, so I'm webbing in from Lafayette, Indiana, currently. But when I visited this past September, I was able to meet a few of you weed control experts. So with that, I have a short noxious weeds update. Just briefly introducing myself. Going through some of my previous research and then what I envision for this position and how I can serve the people of North Dakota through my research. So next slide, please. So just again, a brief introduction, kind of a little bit about my background and then what landed me at North Dakota State. And then get into some previous research. I won't get too in depth there, but just kind of highlight some of my previous experiences. And then lastly, just a few slides talking about where I envision my program going. So first, just a quick about me. So I just really love spending time outdoors, whether it be fishing, playing golf, or disc golf. I love camping and hiking with my girlfriend, as well as attending collegiate athletics. I'm definitely looking forward to rooting for a highly competitive football team. So Purdue hasn't had much success in that realm. Also looking forward to fishing out in North Dakota there. So largely I'm a bass fisherman out in Indiana, but I'm looking forward to targeting some new species out there. Next slide, please. So I grew up in Clear Lake, Iowa, and that's where I was born and raised. It's about 5 hours from Fargo, just in that north central part of the state there. I come from a very strong farming heritage. So both sides of my family have been farming for well over 100 years. And the bottom left, you can see a picture of the homestead my father grew up on. And so that's actually been in our family for over 150 years. It's originally purchased directly from the United States government. In the center there is my mom's dad, so my maternal grandfather, with his first corn crop in 1950. And you can see how proud he is, that was knee high by the 4th of July. And then a little closer to today on the far right is my younger brother, who's the 5th generation farmer, teaching me how to operate his combine. So I have, a pretty strong farming background. And that's kind of what led me into pursuing agriculture in school. And so on our next slide here, just my education, where I did my bachelor's degree in horticulture at Iowa State University. It was there that I worked with Dr. Nick Christians, and he kind of opened the door to grad school. And so I did my master's and PhD both at Purdue, working with Aaron Patton, who's the turfgrass weed scientist. So if you guys don't know, actually Dr. Joe Eichley also, did his PhD at Purdue. So I know Joe from his time here, but I was in a little bit different lab and had just a little bit different experience than what Joe did. And so shifting gears kind of about my background and moving more into some of my previous weed science research. And so I'll just talk about some kind of cool trials that I've worked on through my years in grad school. And so first off, just looking at a project that was looking at the nitrogen application timing effects on mesotrione activity. And so mesotrione is the active ingredient in Callisto or tenacity in turf. And there was some research showing that if you couple your mesotrione application with nitrogen, that you can enhance your efficacy. And on the right here, you can see that the bleaching symptomology that's characteristic of mesotrione. And so what I did with this project is we investigated nitrogen application timing and how that affects mesotrione. And so we applied 43 pounds of nitrogen per acre. In turf, this is 1 pound per 1,000 square feet, which is a fairly common nitrogen application rate. And then we applied that nitrogen 14, 7, 3, and 1 day prior to our herbicide application. We applied nitrogen immediately before that mesotrione application, and then we followed nitrogen up with 1, 3, 7, and 14 days after that mesotrione. So again, just trying to figure out, is there a nitrogen application timing effect on that mesotrione activity? We used the 5 fluid ounce per acre mesotrione rate, and we applied this to 3 to 5 leaf large crabgrass. And so what did we find here? We can see that on the next slide. And we found that nitrogen applications 3, 1, and 0 days before mesotrione application had the highest percent of bleached and necrotic leaves. And you can kind of see that on the graph, or on the figure on the right. And I'll get into that a little bit more. But first, I just want to show you visually what we were seeing there. And so first, we have a picture here on the next slide of some treated plants shortly after, just a few days after mesotrione application. And on the left, you can see a crabgrass pot that received fertilization 7 days before mesotrione. On the right, 3 days before mesotrione application. And it was here at that 4 day after mesotrione application that we started seeing a little bit of symptomology, more symptomology in those plants treated right around the mesotrione application. You can kind of see that in that red circle there, just a hint of some bleaching symptomology there. And so fast forward a little bit on our next slide here, and you can see progressing a little bit that we're getting more activity in that 3 days before application versus 7 days. And so this is 7 days after mesotrione application, and not only are we seeing more bleached leaves, but it looks a little more stunted there as well. Next slide, please, and we'll see a photo 21 days after mesotrione. So now we're moving from some of that bleach symptomology into necrosis. And so again, you can see that 3 days before application is it had more bleached leaves. Now it has more necrotic leaves, and that percent control is higher as well. So on our next slide, I told you we kind of revisit this figure here. And so I think it's important as a researcher primarily to ask the question, well, what does this mean for practitioners? Well, in the example of this experiment, within that red box there on the right, we can see that if we pair our nitrogen and our post-mesotrione applications together or in proximity, we can enhance our crabgrass control. So in this specific case, that may be a golf course superintendent or a sports field manager that's just trying to schedule when their nitrogen applications and when their herbicide applications should be applied. And so this is just something to consider there, that we can kind of enhance control if we pair them together. So on our next slide here, it is kind of the crux of my PhD work, and I'll be speaking a lot more about this plant here, this buckhorn plant. But during an earlier talk, when Philip was asking about 2,4-D overuse, I thought that was quite a timely question that he didn't realize he was asking. And this is the result of 2,4-D overuse, right? So on the paper on the left is the first report of this 2,4-D resistance in buckhorn plantain. And for those of you not familiar with buckhorn plantain, it is a turfgrass weed. It's in all 50 of the United States. It's a quite widespread weed, but it's not necessarily impactful in row crops. So mainly it's a turfgrass weed. But again, my advisor found a 2,4-D resistant biotype in a cemetery in kind of central Indiana. And upon reporting this to the people of the state, we found that, hey, this wasn't the only instance of this. And on our next slide, we can see that in the top left, we found a susceptible type, but that center top one was that original eco type of resistant buckhorn. And we have since found a total of 5 that we have confirmed to be resistant, but there are I have little doubt that there are more out there. And so I guess just to revisit, so all of these plants receive 10 times the normal labeled rate of 2,4-D. And so that all these resistant types are able to survive a pretty high labeled rate. And you can see in that comparison to that susceptible in the top left corner that this is definitely a deadly dose on normally susceptible plants. And so when we consider where these are coming from, it's all turfgrass systems. And so next slide, please. We'll see that these plants have all originated, these resistant ecotypes have all originated in pretty diverse turfgrass systems. So just these confirmed ones, we found them in cemeteries, home lawns, sports fields. We have there's reports from Pennsylvania of them being on a golf course. I've actually collected another type from a golf course that we suspect to be resistant. And so I think this was a more widespread issue than what we even knew. And it's just now that since we've identified it, there's other people coming forward saying, hey, I'm having trouble controlling this weed with 2,4-D as well. And if you're familiar with this weed and herbicide labeling, you'll know that when you get into a home lawn situation, 2,4-D is essentially your only chemical control option labeled for that situation. So if you run into a 2,4-D resistant buckhorn in a home lawn, your chemical control options are essentially nothing at this point. So we took these resistant types and then we looked at, well, there's obviously a mutation here. Does that mutation in the plant affect how it grows in other ways? And so we did a field study, a garden study is what they're called specifically, looking at that. So we took those 5 resistant types and then 7 susceptible types and measured all sorts of different things to figure out, do these resistant types have any morphological differences or any reproductive differences than the susceptible types? And what we see on the next slide is that while there were some differences between these, we weren't able to directly link these to 2,4-D resistance. So we did see some differences in winter survival, flowering date, if they grew upright or more prostrate, differences in their leaf morphology as well. But again, we didn't directly link this to 2,4-D resistance. So maybe there aren't any fitness penalties. Maybe there's just different mechanisms of 2,4-D resistance. So there's many complicating factors, and we did a lot of work with very few answers on this. And so we kind of took a different route in the greenhouse then and trying to figure out, well, what is happening inside these plants that's allowing them to survive these 2,4-D applications. And so I won't get into the organic chemistry involved on this slide here. I just want you to see in red there that cytochrome P450. So cytochrome P450 is an enzyme that can break down herbicides within the plant. And you can see on the bottom there, on the bottom of that right, it says tolerant species. So this is what we hypothesize tolerant species do with 2,4-D. They're breaking that down with their cytochrome P450. On the left side of that, we can see what happens in these sensitive species. And what I really would like to highlight is that double arrow. It goes both ways, right? So this is why they're sensitive. So once you apply 2,4-D, those sensitive species, they don't know how to break that down to a non-lethal form of 2,4-D. And so it kind of flips back and forth. And what's important is that center molecule is the active form. So anyway, what we did is we applied malathion, which is a common insecticide, but what it does is it inhibits that cytochrome P450. And so what the idea here is, if we can inhibit cytochrome P450 and reverse these resistant types back to susceptible types, we're going to get a clue into how that resistance works. And so on our next slide, we can see that when we applied this malathion 18 hours prior to 2,4-D, we saw differences in early symptomology for these resistant types, but we did not see differences in control, fresh weight, or dry weight. So again, we saw some hints that they were different early on, but that wasn't sustained. And there's actually some other research in other 2,4-D resistant species that kind of supports that this malathion has a little bit of an effect, but not a full reversal. And so I'll go through some pictures here of kind of showing what this is. So I'll give you just a second to break down this matrix here. So along the top, we have our progressive picture. So 0 days after 2,4-D treatment, 4 days after, and then 7 days after. And these pictures are all that at a 5 times labeled 2,4-D rate. The first two rows there are produce susceptible. On the top, they did not receive malathion. On that second row, they did receive malathion. And within that susceptible population, we didn't see much difference, or actually we saw no difference between malathion or no malathion. However, in that resistant type in the bottom 2 rows, we do see an effect there. So on the without malathion, you can see very little epinasty or other symptomology of a synthetic auxin. And then on the bottom where we applied that malathion, you actually see a difference between those two. However, this was just early on and it wasn't sustained. So we're not quite sure what's going on there, or if the malathion or this cytochrome P450 metabolism is only part of the puzzle. And so on the last slide, you can kind of, or in this next slide here, you can see that bottom right-hand corner is what I really want you to look at and compare it to the picture immediately above it. And you can see that in that resistant type, when we apply a cytochrome P450 inhibitor, we're getting a little more symptomology there. On next slide here, we can see again at that 5 time labeled rate, 5 days after treatment. We can see again on that right side, when we're putting that malathion on, we're getting a lot more epinasty, a lot more symptomology. Next slide here. So real quick, I know I'm running short on time here, but I do have some non-chemical control options that can influence this buckhorn plant and persistence and viable seed production. And so on the next picture here, we have one of my research plots. And this was looking at, well, how does mowing and nitrogen fertilizer reduce buckhorn plant? And so this was a factorial design in that we had 3 mowing heights and 4 nitrogen rates. And you can kind of see that laid out. And so every mowing height had all the different nitrogen rates. And so on our next slide here, we can get into some quick results. And we saw that mowing height, as we increased our mowing height, we also increased our buckhorn coverage. And so we found that lowering our mowing height helped reduce our buckhorn. And then in the right, we can see that as we apply more nitrogen, we are reducing our buckhorn coverage. So it's some interesting results here for sure. And if we can go through some of these pictures, we can see that low mowing reduces buckhorn coverage, but at a cost, right? So in the center there, we can see that there is no crabgrass, and everything around it in that low mowing height is all crabgrass. So again, we can reduce buckhorn, but we're just trading off our weed species. We saw that nitrogen fertilization reduces buckhorn, and it just so happened that our no nitrogen was right next to our high nitrogen plots. And you can see in that right, even in this kind of pixelated picture, you can see that there's more buckhorn in the low nitrogen versus the high nitrogen plots. Next slide, we can see that this was an effect also on dandelion. So you can see there's fewer dandelion plots, or there's fewer dandelion in the high nitrogen plots. And then we also looked at, well, can we mow frequently enough that we are cutting those seed heads off before they produce viable seeds? And this was an interesting study because we found that no matter how frequently you mow, it produces the same number of seed heads. It's just a matter of, is that seed viable or not? And so what we found was when we germinate the seeds that we harvested off of those plots, we found that sure enough, if the more frequently you mow, the fewer viable seeds that are produced. And especially when you look at mowing every month versus every 2 months, you can see that mowing every 2 months had the highest viable seed production. And part of that was you can see that they're producing much larger seed heads, and so it's just producing a higher seed yield. And then another kind of cool project I was involved with was using digital image analysis to count dandelion blooms. And so on the left, you can see a dandelion plot. We took a picture of it, and then we were able to code and figure out how to use that digital image and count those dandelion blooms using, this software. And you can see on the left is the actual picture. On the right is what our digital image analysis found and counted as a dandelion bloom. And if you zoom in really close on some of those dandelion blooms that coalesce together, our program was actually able to separate those and count them individually. And so this was a cool side project I was able to participate in, and definitely something I'm interested in dabbling with in the future. So where am I going to take my research program? I just got a couple more slides here. So again, there's 13 noxious weeds in North Dakota. And so what's unique about this position is that normally you are researching weeds in a cropping system. Well, it's kind of reverse for me, where I am investigating these specific weeds no matter where they show up. And so one way I'm going to do that is investigating the weed biology and ecology. And so as a turfgrass scientist in training, I look at these 13 noxious weeds, and few tolerate repeated mowing. And so in rangelands and pastures, roadside ditches, oil fields, maybe we can introduce mowing as a control method. I think it's also important to determine the influence of management practices, figuring out what can we do in terms of non-chemical control options. And then lastly, looking at some of the genetic diversity of noxious weeds in North Dakota. I think this is going to be especially important when we're looking at palmer, figuring out, what are these different populations of palmer resistant to. I would suspect that there are going to be multiple resistant issues there. And then the idea here is just to identify what weaknesses these noxious weeds have, and then try to exploit them. And so just taking a system-wide approach, I'm running short on time, so we can just kind of paddle through the last here. So I'd like to explore some weed control technology. So kind of what I talked about with using digital image analysis to count dandelion blooms, try to adapt this to sensing, weed sensing technologies. And I kind of feel that, if some amateur coders in my lab can kind of figure out how to do this, that maybe the right collaborations will open the right doors and enable us to do some of this. And then also looking at some of these new herbicides and figuring out where we can use them. And then lastly, this is definitely something I'm going to have to collaborate with, but so looking at how does noxious weed legislation affect those noxious weeds. So there's some counties that list black henbane as noxious, and I'd like to work with maybe a social scientist and figure out, how effective are these county noxious weeds legislative, and then maybe even at the state level. And so there's some interesting things there to look at, more of the social side of things. Obviously, I'd be doing the weed science portion of that, but this is just something that I could see as a potential collaboration, looking outside of ag. And then I think that should be it. Yep. So this is my NDSU email. I've been having a couple issues with it, so just maybe save this for once I arrive on campus. But I'm very much forward to looking forward up there, to moving up there and working with you all. I'll put through. I can see some of the questions here. Jason, thank you. Yep. I actually have a lab member that he did his undergrad at NDSU, and so he's very clear that it is pronounced bison. So I appreciate that. So Claire asked if I have any interest in grazing noxious weeds with goats, and yeah, I think that's definitely an avenue to explore here. trying to figure out, the efficacy of some of, grazing and mowing, and maybe even directly comparing those two things would be beneficial. And it's working with legislators as well, and county weed commissions and things like that, just to figure out, well, what are these issues? How can we best combat them? What's working in the field? And so this is definitely a new opportunity for me, and I'm very much looking forward to it.
All right. So, Quincy, once again, thank you. And I see a couple other comments in the chat box. So if you want to interact with some of those folks, I'm sure they'll be happy to. And again, we're excited to get you up here and just practice on. It's Bison with a Z. Jason Hanson will personally track you down and correct that for you if you don't get it right by the time you're up here.